Hi, today we're going to cover Chapter 4, which is our carbon chapter, to set us up for some of the things that we need to learn in Unit 5 about macromolecules. First, we just want to talk about what organic chemistry is. Organic chemistry can sound scary, however, it's simply the study of any carbon that contains carbon. Just as a reminder, we've got carbon over here on the side. This is carbon right there, that black center atom. Carbon has a valence of 4, meaning that it can bind to all of our other really important, biologically important molecules. So it can bind to carbon, hydrogen, oxygen, and nitrogen. Remember, a valence of 4 means that carbon is going to want to participate in up to 4 bonds to fill its outer valence shell. As we see in Chapter 5, things like proteins, DNA, carbohydrates, even lipids are all composed of carbon compounds, so it's important to understand how this molecule works. One of the first things that we introduce in this idea is, where did we get the first organic molecule? So where did the first DNA molecule come from? Where did the first carbohydrate come from? Where did the first DNA come from? And the hypothesis is that early on in Earth's atmosphere, just because of the interaction between cosmic radiation and the inorganic elements that were already here on the surface, that they just bumped around until they created organic compounds. Stanley Miller, no relationship to Professor Miller, was a scientist who wanted to investigate this particular hypothesis. So what he did was he set up the following situation where he simulated or recreated a primitive Earth environment. He made sure that there was water in the apparatus, because we know that the early Earth had seas. He then allowed this to get warm so that it would go up into water vapor, think it would turn into clouds. He would then allow it to mix with what we know were some of the compounds in the early Earth's atmosphere, and things like ammonia, methane, and hydrogen. He would then zap this with an electrode to represent some of that energy that was being bombarding the Earth at that time. And then he would pass that vapor through a condenser to cause that water vapor to rain, and then he would just continue this process over and over again. After doing this for some time, he took out a sample for chemical analysis, and in this chemical analysis, he found organic molecules. So even though the things that he put in weren't organic, they weren't DNA, they weren't carbohydrates, they weren't amino acids, when he took out his chemicals to analyze them, he was finding amino acids or building blocks of DNA. So what he shows us is that you can synthesize organic compounds with nothing being alive, right? He didn't need any cells, he didn't need something to already be alive in order to get some of these more complex organic compounds. In concept 4.2, we just again review some of the concepts we've already seen in earlier chapters. First, we want to remind ourselves that it's the electron configuration of an atom that really determines the type and kind of bond that an atom will form with other atoms. So let's think about carbon. We know that carbon's valence is 4. It's going to want to make 4 bonds. It's not going to want to make van der Waals interactions. That has nothing to do with carbon. It's not hydrogen, so that is not going to be a carbon bond. Is it an ionic bond? Well, no, it's not our single example. So when we think about the kind of bonds that carbon can make, what it's doing is it's making 4 covalent bonds. So again, those are our strong shared electron bonds. Carbon, because it can form these 4 covalent bonds, it's able to make very large, complex molecules possible. Each carbon bonded to 4 other atoms has a tetrahedral shape, which is that sort of pyramid shape that you might remember from one of the earlier slides. However, when 2 carbon atoms get joined together by a double bond, those 2 carbon atoms actually get smashed out into a single plane. And you can see that well on the next slide. So here we have a single carbon atom bound to 4 other atoms, a carbon bound to 4 hydrogens. That would be an example of that tetrahedral shape. If you stick 2 carbon atoms together, sort of in the second picture, you can see it looks sort of like 2 pyramids stuck together. However, when we start using a double bond to hold carbons together, that molecule gets a little bit flattened. Please do not worry about memorizing these different molecules and having to memorize these names. These are just being used as examples, so you do not need to have to draw ethane on an exam or something like that. Because carbon has that valence of 4, it's going to be compatible with different elements. Here are the valences that we've been talking about. Just as a reminder, the valence of hydrogen is 1, the valence of oxygen is 2, of nitrogen is 3, and of carbon Our valence is 4, meaning that each of these different atoms is going to want to make that many bonds to complete its valence shell. Carbon atoms can partner with atoms other than hydrogen. For example, here's a great one, CO2, where you can see the carbon binding with oxygens. Down here in urea, you can see it binding to nitrogen. Again, these are just being used for illustration purposes. You do not need to memorize these particular molecular formulas. Because carbon has that valence of 4, it's very diverse molecule. It's able to form lots of different structures and shapes. What we find in biology is that carbon chains form what we call the skeletons of most organic molecules. So when we're talking about DNA or lipids or proteins, those molecules are going to be made up of carbon serving as sort of that backbone, and then other stuff stuck onto it. Carbon chains can vary in their length and shape. We can look at double bonds, single bonds, straight versus branched. In the next couple of slides, I'm going to go through examples of some of the various shapes that carbon may take. Again, these are only for illustration. You do not need to memorize any of these shapes. We're just trying to show you some of the versatility that carbon has. So, on our first one, here you can see we've got ethane versus propane, sort of a 2-carbon molecule versus a 3-carbon molecule, so it's easy to get long strands of these. Next, here we have a straight versus a branched form. We've got butane versus isobutane. So here we have those 4 carbons in a straight row versus over here where those carbons are branched. You'll notice in both of these that in addition to carbons, we just have hydrogens bound. Here you can see the double bond, right? So we can put the double bond on the end, or we can put the double bond in the middle, and that's going to make these molecules different molecules. Again, those are just carbons and hydrogens. Carbons and hydrogens can also form ring structures, and we see these often, especially when we look at carbohydrates. So here's a very common structure, cyclohexane, where you get 6 carbons in a ring. When you see these little hexagon structures, they're just trying to show you that carbon skeleton. If you notice on this picture, like in the last previous slides, all of our elements or compounds that we've been looking at are carbons plus hydrogens. There's a special name for any molecule that is just carbon plus hydrogen. These are called, not very creatively, hydrocarbons. So everything that we've just looked at in the previous slides, they are hydrocarbons. They are organic molecules that consist of carbon and hydrogen. These are a major component of petroleum. Fats have a lot of hydrocarbon components. Hydrocarbons are a useful molecule because they can store and release a large amount of energy. When we think about hydrocarbons, they are hydrophobic. Remember, hydrophobic means that you are water hating. Hydrocarbons do not have any charge on them. They share the electrons very equally, and so because of this, they do not like interacting with water. Another thing that we talk about with organic compounds and organic chemistry is something called an isomer. Please be careful with this word not to get it confused with the term isotope. Isotope was back in chapter 2. That's where you have the same number of protons but different numbers of neutrons. Now we're talking about this thing though, these isomers. Isomers are compounds that have the same molecular formula but different structures and property. So there are 3 different types of isomers. We'll look through images of each of these. Structural isomers have different covalent arrangements of their atoms. Usually it's a straight versus a branched form. The number of potential isomers is going to increase as you get more and more carbon atoms in the skeleton. There are cis-trans isomers. These are isomers that have some difference around a double bond. They differ in their spatial arrangements around a double bond. And then we have enantiomers. Enantiomers are isomers that are mirror images of one another. So let's look at structural isomers. If we look here at pentane and we write its molecular formula, it has 1, 2, 3, 4, 5 carbons, so C5, and then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogen. So its molecular formula is C5H12. If we look at 2-methylbutane, it also 1, 2, 3, 4, 5 has 5 carbons, and if we count up the hydrogens, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 has 12 hydrogens. So these molecules have the same molecular formula. They're both made up of 5 carbons and 12 hydrogens, but they vary in their spatial orientation. So we call these structural isomers. Next, we have cis-trans isomers. You can see that there is a double bond. That's a giveaway that we're talking about a cis-trans isomer. So there's a double bond, and then the two somethings either happen on the same side of the double bond, in which case it's called a cis isomer, or they happen on opposite sides of the double bond, in which case it's called a trans isomer. You've probably used this terminology without realizing you were using this terminology. When we talk about trans fats in our diet, these are fats that have this trans orientation around a double bond. The third type of isomer is an enantiomer. These are mirror images of one another, sort of like a left hand and a right hand. No matter what I do, I can't flip my hand around to be an exact copy of the other. It's as though there was a mirror going through the middle of them. The same thing happens with molecules, and we have what are called left-handed and right-handed forms of molecules. Enantiomers become really important in the pharmaceutical industry because biological systems are often very easily able to differentiate between the two molecules. And so two different enantiomers, just like your left hand and your right hand are different, two different enantiomers may have different effects. Usually it's the case that one isomer is active and the other is inactive, but that's not always the case. So we want to remember that organisms are going to be sensitive to even subtle variations, just like being able to tell the difference between a mirror image. One example of this is the drug thalidomide. Thalidomide is a drug that was developed as an anti-morning sickness drug. It's really good at helping prevent morning sickness. However, it comes in an enantiomeric form. So one form is great at curing morning sickness, and the other causes limb malformation during fetal development. So you can see this kid over here, right? His mother unfortunately was taking thalidomide, and that child suffered. A couple examples of things that you might be more familiar with, things like ibuprofen and albuterol. They have both an effective enantiomer, that thing that actually does whatever you think that the drug does, and then there's an ineffective enantiomer. This is often the time why on the outside of your ibuprofen bottle you may see something that says this is 100 milligrams of active ingredient. The pill itself might actually be 200 milligrams, but they know that half of that is the ineffective form, so they're only giving you the 100 milligrams that's effective. So those are the three different types of isomers. You should be comfortable with what an isomer is and then be able to differentiate between structural, cis-trans, and the enantiomers. Concept 4.3, we're going to talk about functional groups. Organic molecules really depend not on that carbon backbone, but the extra things that we add to the carbon backbone. Those extra things that we add to the carbon backbone are called functional groups. These are molecules that are most commonly involved in chemical reactions. There is a large list of functional groups that you can learn about in chemistry classes, but for biology purposes, we're going to learn about 7 functional groups, and we'll be using these functional groups throughout the rest of the semester. These functional groups are going to give each molecule its very unique properties. So I just want you to think for a second as we look at these different pictures, which one's the boy and which one's the girl. So here's our male lion, here's our male peacock, peahen, and then here's our male mallards, and those brown ones, those are the females. And if we think about what makes a difference between these male and female animals, really it comes down to estrogen or testosterone. So what I want to do is I want to show you what estrogen and testosterone look like. Down here at the bottom, we can see estrogen and testosterone, or estradiol and testosterone. They're both steroids. They look really similar, right? Hexagon, 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 pentagon. Hexagon, 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 pentagon. In fact, the only difference between estrogen and testosterone are those places that are highlighted in blue. Those parts that are highlighted in blue are examples of functional groups. There are 7 functional groups that you will need to learn about. They include the hydroxyl, carbonyl, some people call it the carbonyl group, the carboxyl group, the amino group, sulfhydryl, phosphate, and methyl group. For all 7 of these functional groups, you need to know that they are hydrophilic. Again, hydrophilic means water-loving. So if you take these and you stick them on an organic molecule, if you stick them on a hydrocarbon, something that usually doesn't like water, now, because this thing likes water, it's going to help make that organic molecule more likely to be soluble in water. For each of these functional groups, you should be able to do the following. Number 1, you should be able to name it. Number 2, you should be able to draw it. Number 3, you should be able to name compounds that contain it. And number 4, you should be able to tell me some important functional property of that functional group. So here's our hydroxyl group. If you're drawing a hydroxyl group, it's drawn as an OH group. Hydroxyl groups are important because they are going to be polar. And if you name a compound that contains a hydroxyl group, these are your alcohols. Next is our carbonyl group. The carbonyl group is a carbon double bound to an oxygen atom. The carbonyl group is important, especially with our sugars or carbohydrates, because it makes things act as an isomer in many cases. We use it to create different isomeric forms. We name carbonyl groups based on where the carbonyl group is found. If the carbonyl group is found in the middle of the molecule, we identify that as a ketone. If the carbonyl group is found at the end of the molecule, we identify that compound as an aldehyde. The way that I remember it is in my family, I'm the oldest and I'm Gwen. And then I have a sister named Katie, she's in the middle. And then my baby brother, his name is Adam, he's at the end. So I remember it, hey, Katie, the ketone, she's stuck in the middle. Adam, my baby brother, he's stuck at the end. Again, you can use whatever you want to do to remember how to name compounds that contain carbonyl groups. The next group that we have is the carboxyl group. The carboxyl group is actually going to look like a combination of the first two. So in a carboxyl group, you have a carbon double bound to an oxygen, sort of looking like a carbonyl group. And to that same carbon, you're going to have a hydroxyl group bound to it. So it's called a carboxyl group. These are important because if these get stuck on molecules, they act acidic. So this is going to make molecules more acidic. To name compounds that contain carboxyl groups, they're called carboxylic acids. The next functional group that we have is the amino group. The amino group, try to listen to that N sound when you say it, is the only one we're going to talk about with a nitrogen in it. So the amino group is a nitrogen bound to 2 hydrogens. These are important because they act as bases, and if you stick an amino group on something, that compound is now called an amine. Next, we have the sulfhydryl group. The sulfhydryl group, focus on that S sound, is the only one with a sulfur atom. It's a sulfur bound to a hydrogen. Sulfhydryl groups are important, especially in proteins, because when you get 2 sulfhydryl groups next to one another, they're going to grab onto each other very tightly and create what's known as a crosslink. This helps stabilize protein structure. When you add sulfhydryl groups to atoms, these compounds are then called thiols. Next, we have a phosphate group. The phosphate group is the only one with a phosphorus atom. It's probably the trickiest one to draw, but it's not that bad. In order to draw it, start with that central phosphorus atom. So that's the phosphorus in the center. Then it's a double bond, single, single, single. Around all of those bonds, you're then going to add an oxygen. So an oxygen, 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 oxygen. 2 of the singly bound oxygens are going to have a negative charge associated with them. Phosphate groups are important because when we see them, they're often involved in energy transformations. If something has a phosphate group on it, we call these things organic phosphates. Our last functional group is a methyl group. The methyl group is a carbon bound to 3 hydrogens. This is important because it affects gene expression. Methyl groups can be added to DNA and they can block genes or they can enhance genes depending on how they're placed. Compounds that have methyl groups added to them are known as methylated compounds. Again, this is the real meat and potatoes of chapter 4. Please make sure that you memorize these 7 functional groups. Know their name, know how to draw them, know what's important about each of them, and then know how to name them when they're added to compounds. On this slide, we talk about ATP, and right away, what I want you to notice is we can see a functional group. Hopefully, you can recognize that as a phosphate group. Phosphates are important for energy transformation. So already by just looking at this molecule, I know that this molecule is an important energy transferring molecule. That's all we really want to talk about ATP at this moment, just so you can see how those functional groups are going to be important in biology. On the next slide, I have a couple of pictures of molecules that I'd like you to look at. So here we have some different molecules, and what you might see on a quiz or an exam is some molecule like this that you don't know what it is, where I ask you to identify functional groups. So let's start with letter A, just look at that for a second and try to see which functional groups we can identify. So right here we have one, that is our carboxyl group. Over here you might also call this a methyl group. Letter B, take a moment to look at that. Letter B is, again, you see that carboxyl group. You also see that carbonyl group. Note that that carbonyl group's in the center, so this would be an example of something like a ketone. And if you wanted to, you could also say that that was a methyl group. All right, looking at the bottom one down here, again, sort of look at it for a moment, see which functional groups you see. I see an amino group down here. Amino groups are going to make things act as bases. I see a hydroxyl group. It's going to make it polar. And I see that carboxyl group, which is going to make it act like an acid. So these are the types of things that you should be comfortable with. Again, please know your functional groups. This will be a concept that we use throughout the rest of the semester.